and welcome to the SquaMates podcast. This is a very serious podcast about herpetology, and I'm one of your hosts. My name is Dr. Mark D. Scherz. I'm the curator of herpetology at the Natural History Museum of Denmark, and I'm joined today by these three lovely people, Ethan Kosak.
Hello. I'm Ethan. I am an artist and illustrator and reptile and amphibian enthusiast.
And Hiral Naik.
Hi, Hiral here, snake ecologist and conservationist from South Africa.
And Gabriel Lughetto.
I'm Gabriel Lughetto, and I'm a scientific illustrator, paleo artist, concept artist, and I used to work in herpetology, but not anymore.
Welcome back, friends. It has been a very interesting year so far. We're recording today on the 8th of May, and there have been some interesting papers. So we're going to dive into some papers. No points for guessing what kind of animals we are talking about today. It seems that this year we've been doing quite a lot of snake talk, and I don't know why that would be. So, yeah. Curious, but anyway, so we're gonna do that. But first, how are you guys doing? What's been happening in the world of all y'all, as they say?
Who goes first?
I'll go first. So I'm working on a new book. We can put the link to the page for it in the show notes, but it's gonna be launching as a crowdfund mid-July.
And it's called?
It's called The Unfamiliars.
And it is a big middle finger to
J.K. Rowling-esque type stuff. Well, her specifically. Yeah.
Yes, I've seen some of the early doodles. They look really fun.
Yeah, they look awesome.
Thank you. Yeah, it has a newt, which is also my D&D character, called Hiral, who's a blind swordsman, sort of Zatoichi character. And he's blind because the sorceress who made him kept plucking his eyes out for spells, and they stopped growing back.
I don't know if newt eyes grow back, actually. Do they?
Yeah, actually, possibly neotenic ones, because I know axolotls can grow back parts of their brain.
I know that they can regrow limb and brain, but I don't know if they can regrow whole fully formed eyeballs.
Yeah. Actually, that's a good point. I don't know. Somebody else told me that eye of newt is in reference to like mustard seed anyway, so it's fine.
Huh. How interesting. Okay, very cool. Gabriel, how about you?
I've been working on a lot of stuff that usually, as always, I cannot.
Yeah, I don't even know why I ask, Gabriel.
Under embargo. It's all under embargo. And let me see. Yeah, that's basically what I've been doing. I've been working on the stuff that is under embargo. But I'm going to say that I'm working on, well, that might come later this year, but I'm working on illustrating something that is super, super cool, and it will blow people's minds.
Ooh, very exciting. Do you have any idea of a date when that might come out?
No.
Okay. All right.
Because I don't think the paper has even been sent yet. So,
okay. All right.
I think I saw you did some nice ground sloth illustrations.
Oh yeah, that was for my licensing library.
Those really nice.
Yeah, I love ground sloth, and it's also always fun to see them depicted, because also they went extinct so recently, just a few thousand years. So it's crazy to think that we had something the size of an elephant walking around here.
In addition to the elephants.
Yeah. In addition to the elephant. Yeah.
How about you, Hiral?
Oh man, the last month has been pretty crazy. I've been trying to get a paper submitted, my first paper for my PhD submitted, which I finally submitted to a journal last week Friday. So it's been kind of hell, but first submission, right?
Well done. That's great. That's great. Can we know what journal you've submitted to?
So this one's mostly focused on kind of the snakebite incidences. So I submitted it to the Royal Society of Tropical Medicine and Hygiene.
Very cool.
Which is pretty exciting.
Yeah. That sounds like just by name alone, which can sometimes be a good way to judge it. It sounds like a tough journal. So fingers crossed for you.
Yeah, it's interesting talking about snakes and from the snake perspective
Yeah,
rather than
right,
the medical perspective.
right.
So yeah, hopefully people find it interesting.
Yeah. Yeah, it's amazing also how much of the PhD process is really focused on those submission like timelines because it takes a surprisingly long period of time to go from a paper that is basically ready to submit to actually submittable. Like all the formatting
Yeah.
things, you have to write the submission letter, you have to make sure that all of the reviewers have gotten the okay, the co-authors are okay with it and everything, and then you have to fight your way through one of an infinite number of submission systems in order to get the things in the first place. It really is a very involved process for those listeners who are not, involved in the academic side of things. I think it's hard to imagine how much work it actually is to get these stupid things even submitted. And then the worst that happens is that they desk reject and you get it back, 6 hours after you've submitted it. You've barely even poured your champagne and you're back to square one, which is really frustrating. So like, yeah, I sympathize with you a lot, and I know, or empathize rather. But I hope that, well, it sounds like it hasn't come back yet, which is great. That's usually a good sign. So
Fingers
yeah,
crossed.
yeah, fingers crossed. And as for me, well, it's been a very intensive time. I've actually spent quite a bit of the last month on sick leave because I've had some jaw problems and some flu problems and a sick kid. So it's been a bit of a rough go of it. But um on the other hand, there is some good things. So we had two papers published actually. One of them is describing four new species of Gephyromantis, little frogs from the Gephyromantis moseri species complex, which I quite like because they're little brown frogs that have spikes above the eyes and they sit along streams and do little churring calls. And my colleague Carl Hutter and I discovered a new species of this species group back in 2016. So that's one of the four new species that we've described here. And then the others are, from across the rest of Madagascar. So that's quite nice. And then the other paper that we had published was a description of a new subspecies of frog. And this is quite an interesting thing. So I mentioned this on the last episode, I think. But to go into it in a little bit more detail, we have this really interesting case where we've got a frog that is known from Western Madagascar, like central Western Madagascar. And then in 2016, I found these frogs in northern Madagascar, 200 kilometers away. There is no forest. Like there are just tiniest fragments of forest between the southern population and the northern population. There's a strong mitochondrial difference between these things, a relatively weak nuclear difference between these things, almost no morphological or practically no morphological differences whatsoever, and we don't have calls. Now, normally for describing a species, the call would be the nail in the coffin. There's a high chance that a species that has a genetic distinction, the mitochondrial distinction as high as these have, which is like between 4.8 and 6.6%, I think, in the 16S region that we use. 3% is the cutoff for most species sister pairs. So that's quite a high thing. Normally, we would find strong bioacoustic differences, but we don't have the calls from the northern population. And so we're facing this weird thing where some of the data is a little ambiguous, some of it is quite clearly very distinct, and then some of it is more on the level where we would typically be talking about intraspecific. And so this is kind of, from a taxonomic perspective, quite a useful case for us to use a subspecies, because on the one hand, we use the subspecies to recognize that this is evolutionarily clearly on a distinct trajectory. Like this is an incipient species, whether it's completely separated or not until now. we would have to go back and get calls to have an idea really of whether or not they're fully separated. But it certainly is on its own evolutionary trajectory.
Well,
But we
how long ago did the forests in the intermediate? Because I would imagine they were connected at some point.
they're different drainage basins, so even if they had been connected, there would have been a like it's been a very, very long time. So the forest might have been connected within the last 40,000 years, maybe, but maybe also not. We don't really know the paleo climate
So,
and especially
because
paleo environment across there, plus the drainage
if the taxon
basins.
is forest specific or is depending on forest, it has to have gotten
In the
there.
south, they're not forest specific, they're found in karsts. But in the north, they are in the forests. But they're also in, they're terrestrial breeding frogs. So maybe the drainage basin doesn't matter that much. But in any case, it is a case where, in order to hedge our bets, basically, to allow us to have the ability to manage these things from a conservation perspective, to talk about them from an evolutionary perspective, to have the option later to elevate this thing to full species level, we've opted for a subspecies level description at the moment, which does, as I've mentioned in previous episodes, so there was I think it was 2021 paper by Kevin de Quiroz where talking about the change in a little bit of the change in philosophy for how do we approach subspecies, where we talk about subspecies as a difference perhaps in degree rather than a difference in kind between from species, right? So in this case, we're just saying these things have some evidence, but not a super overwhelming level of evidence to indicate that they are species. So subspecies and species under this revised concept are basically the same thing, except that one is, if you imagine this gray zone between one species and two species, where there's kind of a gray buffer zone of who knows, in that gray zone, as things move through it, they have more and more and more lines of evidence that would support them as different species. In this case, we're basically saying this is somewhere in that gray zone that it is sufficiently distinct to say it's on its way, but not necessarily completely distinct, and therefore subspecies is used rather than species, which would be further along that splitting process. So it's a
Yeah, we have.
utilitarian thing where we're still talking about these things as being evolutionarily distinct entities. So
Well,
Corvus bacillana, which is the new subspecies, is an evolutionarily distinct lineage from Corvus corvus, but there are some things that are not as far diverged as we might expect from things that were recognized at full species level.
well, we have to remind everybody that these are arbitrary lines anyway,
They're
and
semi-arbitrary lines.
well,
Yes.
to a point, because we decide where we make the cut lines. You know what? Where we did, because it's all a gray zone, and we see that in paleontology clearly. I mean, when you go back to the origin of dinosaurs, it's a mess because
Yes.
they all look the same. All the clades look the same because they are basically starting to diversify. And when you were starting to diversify, you basically look the same to your ancestral species. So
Yes, well, yes and no. I think that in the case of modern things, we have a much more to work from, right? And so the way that we can talk about species in the paleontological record, as someone who works on the modern things, it's very difficult for me to conceptualize how the paleontological people actually do this, because when you have a modern taxon, you really have a wealth of data that you can use, and you can really do this integrative taxonomy thing where
of
I
all
can
lines of
look at
evidence.
all of these different lines of evidence.
Yeah.
Whereas otherwise, you might just have a bone. And what is your line of evidence? You have essentially one line of evidence plus time,
Well,
or plus time and
but even
geography.
but what I'm saying is that even with the many lines of evidence that we have when we work on modern taxa, it's still, we have to make a decision whether
Yeah,
you're
no
going to
doubt.
consider it a species or not based on how many lines of evidence. And I
Exactly.
think that that's happened to any of us who have worked with, elevating subspecies to species taxa. Like, that's why I'm not a huge fan of the subspecies concept, because in a lot of times, I think it muddies the water a little bit. But I understand why you did it, and I think it's a very conservative and good way of
I mean,
doing it.
I think that there's a paper by Christoph de Fresnes and colleagues that was published, I believe, in PNAS, where they discussed basically recognizing the, evolutionary diversity without overestimating. So I think the risk is that by recognizing every single one of these lineages that has like some indication of distinction, but not overwhelming evidence of distinction as full species, really would muddy the waters. Whereas, hedging your bets a little bit and saying, okay, some of these are not as distinct as others, it also gives you a little bit of a finer level of distinction. But of course, this only works in a case where the entire system has been revised by people who share that philosophy, because it would not be compatible in a group where some people apply it one way and some people apply it, let's say, the classical way of doing subspecies, where subspecies was, in Darwin's time, essentially just a means of recognizing. That was often used for geographic subunits of different things. There's essentially locales, which have turned out to be, in most cases, crap. Like sometimes there is lineage separation related to those biogeographic things, but oftentimes that was not the case. And
We can
so
blame
a
ornithologists,
lot is
basically.
yes. Well, I think the ornithologists have some very interesting points, and they have been to a great extent more thorough and more careful than herpetologists ever were, because they're the people who were doing early ornithological taxonomy were more thorough thinking and systematic and had a broader overview of what they were working on, which is why you have that beautiful conformity of the things like columbiformes and they have all these different groups that end in formes and
Well,
all of
yeah, but
the
also when they're describing a subspecies, I mean, I find ornithological descriptions terrible in many
yeah,
places because
this is true. So the modern ornithologists do use subspecies, let's say, more liberally than herpetologists tend to. But
terrible.
that's
You
part of the
know,
reason that it's so difficult to compare between these different groups, because it's seldom one person who works on all of them or even reads multiple different groups of taxonomic papers.
they're just ornithologists are just herpetologists with specialization. So
I
uh,
mean, it is true, but that specialization is sufficient that we seldom overlap in literature. And since the very, very earliest days of taxonomy, it's been
yeah,
very hard
I
for
just I want to.
reconciliation.
So I was gonna say, as a lay person with only some understanding of this, it puts me in mind of all the talk about ring species that you used to see. Like, I know that that's been sort of discredited and not, that all the examples that were brought up turned out not to be
Yeah, that's
so
true.
great examples.
Yeah, there
But
are some very nice herpetological examples of ring species, but
it makes me think of
Yeah,
that with this
most of them have
gradation
been
of,
most of
yeah,
them have been
problem.
debunked. Yes,
right,
Yes.
yes.
right. And I know that, but it did, but the idea of gradation between a gray area between species, that's what it makes me think of. And
Yeah. And
But
uh,
many
even
of those cases might have been better appreciated really at subspecies level, because they,
right,
again, it's a case of degree and whether or not these things are fully on their way to, like, obviously we can never be we cannot we are not the arbiters of whether or not these things do go through with the speciation process completely, and some things stay in that gray zone for millennia. So it's not to say that there is always this
yeah.
that every criterion must always be satisfied. I think that's one of the beautiful things about this, unified species concept, where you can appreciate that you can have species that are appreciably totally distinct from one another. I'm so happy it's you, Gabriel, this time, and not me.
Congratulations, Gabriel.
Uh,
I don't know what happened,
and,
but
and
for the
congratulations.
for
especially
the
when we're
listeners
talking
at
about a
home.
subject, especially when we're talking about a subject which I really dislike. So great.
Uh, for the listeners
By
at
the
home,
way, I think that when you have clinal variation,
Mm.
which is often the case, that doesn't show you anyway that subspecies, because there is, you know, there could be genetic flux between two populations, and they still very well be different species. That has nothing, which is the old
Right.
concept of
Right.
subspecies.
This is what I was saying. I think that one of the really one of the things that I most appreciate about this unified or generalized lineage species concept is the very fact that you can have like you can better appreciate the fact that not every lineage must satisfy the same criteria, must have the same level of distinction. So they don't have to be biogeographically separated if they are separated on the other things, or it's not always going to be the case that they will be, in total genomic isolation, because there will still be some gene flow, but some areas of the genome are sufficiently resistant to gene flow that these two entities can persist with ongoing gene flow for a long period of time. We see that a lot in butterflies, but there's also dart frogs where I think that's been shown. So there's quite a lot of benefit that comes with that, but also I think there is this part of that concept is built in to in the early stages of speciation, fewer of those criteria are going to be satisfied, right? Biological species criterion, which is that there's no possibility for gene flow because they're reproductively isolated, that tends to be one of the last things that actually gets fully supported. Sometimes it happens really early, and then everything else happens afterward, but typically quite quickly, because the inability to have gene flow that comes from that reinforces a lot of the processes really, really early. But it can also happen relatively late. So you can have bioacoustic differentiation, but later on get the biological speciation, biological species criterion satisfied, because initially it's sufficiently filtering for the things to only be finding their mates based on the call that gene flow is substantially reduced. It's not that it completely goes away, because mismating happens because frogs are promiscuous, or slash they're very bad at recognizing which partner is which when it comes up to face. And then
basically, they mate with whatever they have in
right,
front of them.
exactly, right. They'll mate with an orange if it stays still long enough. And and later on, you then get that reinforcement that leads to the total biological isolation. And so my point is that you can have, because it happens in a different order in different groups, not all of them are necessarily at all satisfied in different groups. So fishes, for instance, can continue to cross to hybridize 100 million years after they've diversified, after they've separated, given the right kind of opportunity to do so, as we saw between, I believe it was a sturgeon and a paddlefish, that hybrid that was produced, that was totally unfathomable to most people. It's like
cichlids do it constantly.
cichlids do it all the time. You can under the right conditions get an African cichlid to mate with a South American cichlid, which is a bonkers idea.
That
They've
makes
been
no
separated
sense.
for an obscenely long period of time. But they can and will do that if you give them under specific circumstances. And so, yeah, so a lot of these things are more gray than people appreciate. And the way that we are using subspecies in this revised way, we've done this now 5 times. So we've described 5 separate new subspecies in the last 2 years. And the way that we are using it is basically to say, in those early stages of speciation, we think that we have found these cases that are early stages of speciation, which warrant names because they warrant our ability to discuss them in a coherent, cohesive, and easy manner. And we think that they are sufficiently on that path that, given sufficient time, they will go to completion. However, we want to demonstrate by using the name that we have, the way that we have formulated it, to indicate that this is not as far along as most of the other cases that we're looking at. So that's the way that we're using it. It's quite utilitarian, but it allows us, it gives us the freedom of an existing framework that was subspecies that had kind of really lost reputation over the years, and justifiably so, and repurposes it to be a way for us to highlight these particularly interesting cases of incipient speciation. So that's the point. Anyway, that was a very, very long tangent. So after a very brief interlude, we are going to go on to one of the papers that we're going to be talking about, which is the macroevolutionary singularity of snakes. So join us again after the break. Welcome back. Okay, so we are going to be talking about a couple of different snake papers, surprise, surprise. And the first paper that we're talking about is titled The Macroevolutionary Singularity of Snakes, and it's by Pascal O'Title et al. There is
Along
quite
et al.
Along et al. It is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm going to guess around 20 co-authors on this paper. So it is a big group of people, including
Very
some
well-known
fairly
people.
well-known snake researchers, herp researchers, phylogeneticists, etc. So, and of course, this was published in Science. In fact, this was the cover of Science, so it has gotten quite a bit of attention. If you've not heard about it until now, then you must have been under a rock. But that is what we are here for, because we are herpetologists, we turn over the rocks. So let's talk about this paper. It is the first thing that I think I would like to say is that it is such a relief to read a paper about snakes that in the first line says, from a phylogenetic perspective, snakes are indisputably nested within lizards. I would just like to bring attention to the fact that if you go and read a paper by Myreed et al., who have done wonderful things on lizard evolution, but have typically, in their evolutionary analyses of lizards, dropped out all of the snakes, that is, to my mind, an indefensible position, because snakes are lizards, and of course they slightly alter the picture when you start to analyze them alongside everything else. But it's a very important part of lizard evolutionary history that they did this one thing that has been so vastly successful. So I thought that was a wonderful, it is a wonderful preface, and it's also a context for the entire study. So does somebody want to give like an overall, like 20,000 foot breakdown of what is the point of this paper?
Ethan had a good one.
Well, I mean, I was saying before we started that I read through this and I never fully understand anything that I read, but when I did for this, it was kind of like we are seeing snakes as sort of a perfect storm of adaptations and evolution, and beyond that, it's good. It was kind of a we don't know, like.
I think that's so. What I would say is, so the objective with this paper was basically to identify why is it that snakes are so exceptionally diverse, right? That was the kind of the goal, it seems, in the setting out of this paper. But you could actually take that away, and it looks like they tried to frame it this way, where you say, where are the exceptional lineages within lizards? And what has happened within lizards? And that just so happens that snakes really stand out. And so they've analyzed it, or they've presented it largely from the perspective that, okay, snakes are really remarkable, and so let's focus mostly on the snakes. But in reality, this is an analysis of lizard diversification as a whole, which I think is really, that's, as I say, really important to talk about snakes in the context of the lizards.
Am I right in saying that they kind of say that no one particular evolved trait is special when it comes to snakes?
Right. And I think that's, yes, essentially you are. So that, that's jumping ahead to the takeaways from the paper. But I think, that's fine. That is basically the gist, right? So
Yeah,
done,
okay.
all finished. We don't have to talk any further. No,
Okay.
you know,
Well, so before we do that, I just wanted to put people in perspective a little bit. When you are doing, for example, the composition of the herpetological fauna or the reptile fauna of a region, usually in most places, you will have uh, snakes will outnumber non-snake lizards in the same habitat. You will have more species of snakes than other lizards, and that repeats in
in
most places.
especially in South America.
Yeah, but that repeats in most places, I would say.
How about
And
where I'm in upstate New York, and there are no lizards here, but there are plenty of snakes.
Maybe I wonder if that's an American thing though. It's
Oh,
what
like,
about
maybe.
yeah, Hiral, how is it in South Africa? Because chameleons are crazy diverse. The agamids are there. Like, I don't think that I would say that the reptiles, the non-snake SquaMates are still outnumbering the snakes,
Yeah.
but it's a tight race,
Um,
maybe.
it is. I think also Africa is a very old continent, so I think we've got the space to allow both to kind of diversify.
Hmm. So I know in Madagascar, the snake diversity is okay, maybe because of the island effect, although it's hardly an island, um, is maybe poorer. But in general, yeah, snakes are usually outnumbered on those species lists because we've got so bloody many geckos,
but I
skinks,
think
and
that is
chameleons.
an outlier. That's what I wanted to say, because
It's an interesting question.
I don't think the same happens in Australia. I don't think the same happens in Asia. I'm not sure in
Very good
Europe.
question. Yeah.
I'm not sure because in Europe the numbers are so low of everything that I don't know. But, but I
Well,
know
we
for
in Denmark we have two snakes and we have two lizards.
okay,
So
okay.
one for one.
But when you look at stuff like Spain or Italy, I think that
Mhm.
I think they have more snakes. Maybe no,
No, they wouldn't.
they wouldn't, because they have those little islands with all those
Yeah.
lizards. So I
New
don't.
York has Italian wall lizards.
Yes,
But
Bedartzes.
I think
Yes, yes.
in general, and I would bet that probably Africa is the same. I'm not sure, but I would imagine that Africa, even if you have all the chameleons, I would imagine you have more snakes in a habitat. I'm talking about in a region, in a biogeographic region. I also would like to remind people that phylogeny on like taxonomic phylogeny within snakes is extremely underappreciated, because snakes are more conservative, tend to be more conservative morphologically than lizards. And there is a lot of snakes that are differentiating in stuff like hemipeneal characters, stuff that is not visible right away. So just that as a reminder.
I'm really not sure for especially East Africa, just because we like in terms of richness of skink species and gecko species and agamid species and cordillid species and chameleons all
Do we know?
together. I think in the vast majority of places they will be outnumbered, but it's a tight race, and the number of families that can be represented, like you'll often have quite a lot of species, but there's relatively few families that are represented in an area
Yeah,
for the
but
snakes.
that's
But
different, because they even in this
yeah,
paper
yeah,
they talk about the importance of colubriforms and how
yes,
that is like the most diverse within snakes,
hmm
radiation within snakes. So
hmm
that I think that you have to do it on an alpha taxon, like you have to do it in a species.
yeah, yeah, sure,
Yeah,
sure, sure, sure. And you can't compare higher taxa. That's certainly true.
yeah.
But
Well, Haral, your point about Africa being an older and an older continent, that's interesting to me too, because I wonder if you plotted all these things on a sort of paleontological time axis, um, because we're just getting a snapshot in
Well,
modern
but
time.
in that sense, South America is also,
I mean, they're
and
you know
Australia, they're right there.
roughly
Then
the same age.
Gondwana, they used to be together. So
But we've also got really high diversity of chelonians. And I'm actually just looking at the atlas we that was published a couple of years ago on reptile diversity, and I think lizards outnumber snakes in southern Africa at least. And
Yeah.
and I mean, I think it's Mark's point, like, because we've got a huge number of different types of lizards. You know, and I think just, you know, from experience, I tend to, I mean, I see lizards more often than I do snakes.
Certainly the abundance, the detectability and abundance of snakes is much lower than the other lizards because, you know, yeah, but yeah, I do think that it's closer than you might expect, especially given age. So the other thing that comes into it is that snakes as a group, especially Alethinophidians, which is the quote unquote advanced snakes, are relatively young. So one of the things that this paper does, it provides us a new phylogeny of the SquaMates, and one of the things that really jumps out is that the colubriforms is a young and hyperdiverse radiation. Of course, the sampling, there's a little bit of a sampling bias in the tree that's presented because, like, there are well over 1,000 species of geckos, and there are not enough geckos represented in the tree. But on the other hand, you know, geckos are an immensely ancient radiation. And so having a lot of geckos is kind of not as big a deal as having a lot of snakes. So when you take into account age and the age bias thing, I think, Gabriel, back to your point, is like, given their age, there is an excess of snakes in most of these environments. I think that would hold up if you were to do that analysis. Yeah. But anyway, this is far from the point of the kind of the main discussion of the paper. So,
are you suggesting that we got into the weeds again?
yeah, of course we get into the weeds. I mean, that's kind of that's the whole vibe. Hello, listener, welcome to the weeds.
Yeah.
But no, I think the main thing that is so this paper it's kind of typical of one of these science papers. They've thrown so much at the question that to, I think, the lay reader, it's relatively difficult to parse unless you kind of skip the sandwich in the middle.
Lay reader here.
Yeah.
And yes.
Right. So,
Yeah,
and I think, you know, some of their figures are beautiful, but then not necessarily super obvious to interpret. So
Yeah.
I've done in this study some really interesting things, some of which are kind of novel statistical approaches, which is the stuff that science is like, you know, they really like to see people coming at this with an innovative approach, something statistically new. And so one of the things that stands out is the calculation of these of change, like innovation. So they use an innovation index. And the innovation index is a really interesting, curious thing, which is basically it's calculated as the difference between quote unquote the ancestral squamate, whatever that is inferred to be through an algorithmic ancestral state inference, and a given tip. So a given species on their tree. And they do this for various different things. So they look, for instance, at diet. So the inference is how specialized or how different is the diet compared to the ancestor, and then they've corrected this whole calculation for Brownian motion for random drift over time. The specifics of how they've done that, I couldn't even get from the supplements, which is a little bit, yeah. But anyway, so they've done this for various different traits. They did it for diet, elongation, skull morphology. I think there were a couple of other things that I'm forgetting here. So they've done it for
venom, the presence of highly toxic venom.
Yes, exactly. Chemosensory processing, etc. And so you wind up with the really interesting metric that basically is how different is a tip compared to the ancestor, from the ancestor, given the amount of evolution that has had time to take place in between. And then they did a couple of other calculations that are kind of related to this, which is also like how different is one thing from its direct ancestor. So how much innovation is happening along a branch. And this is all very interesting stuff that winds up very clearly and beautifully highlighting the snakes as quite an interesting group. They have this accelerated rate of innovation in various different traits, especially the ones that they highlighted, of course, with diet. Okay, we know that snakes, in terms of their diet, and this, by the way, is based on a surprisingly large data set, over 60,000 natural history observations, diet observations. In terms of their diet, snakes are, they occupy a different space than the rest of the lizards, which I think is something that intuitively makes sense. Gape size change
That's clearly
dramatically.
showing one of their graphics, which
Is
is one of
exactly.
the
It is
best
clearly
graphics.
shown in one of their figures that there is this distinct space that snakes occupy. That yes, some lizards come into some portion of it. There is obviously some overlap, but then there are some areas that only snakes can achieve. So especially eating anything larger than gape size is something that no non-snake lizards have ever really managed to achieve, right? It's because of that elasticity and flexibility within the skull that characterizes especially the Elaphinifidian snakes, especially the colubriforms. And then elongation, which like elongation. Sorry, Ethan, what were you going to say?
No, I was just gonna say like no one. So I was reading that right. No one specific
Yeah, so when you
innovation is really at the ingredient
Well,
for snakes.
I think I think the same thing could be said of any of the lizard groups though, right?
Yeah.
So
well,
if I
that's what one of the points that they make. They do make that point clearly, and it's something we've talked about here in the show many times. Is how many times SquaMates have gone through this, lizards have gone through this process of reducing their limbs, and it has happened in many, many clades. And one of the points they make in this paper is that snakes were the most successful group out of those many, many times where that occurred. Why?
right. Right.
Yeah.
So the point is, it was not the loss of limbs that has led to snake innovation. It was, in many ways, I think you already said this, Ethan, the perfect storm. And I think
Right.
if you look at a lot of other squamate radiations, you would see kind of the same thing repeated. So chameleons, what do chameleons do that's actually different? Like it's actually unique to chameleons. They have a prehensile tail. Well, lots of lizards have prehensile tails. They have independently moving eyes. Well, actually, anoles also have independently moving eyes, and several geckos can also move their eyes semi-independently. They have accommodation cues that they can use for depth perception. That's probably seen in some other things. The tongue is very, very similar to what we see in some salamanders. And so, like, it's not that what we see in chameleons is any feature that is nowhere seen anywhere else. Sorry for bumping the microphone. It's not a feature that's nowhere seen anywhere else. It's the perfect storm. And I think that's what snakes have done is they've made the perfect storm in a way. They combine a lot of these innovations that have led to some interesting stuff. What is, I think, particularly interesting, something that really needs to be talked about, is the early branches of the snake tree. And because we have the early branches of the snake tree, we don't really understand what happens. What happened in very early snakes? Because the surviving lineages that we have from those earliest branches are blind snakes. So the deepest branches within the phylogeny of snakes are blind snakes. We have what are called the Scolophidia, but Scolophidia according to some authors, should no longer be used. According to some other authors, it should be redefined because we have the anomalous, the typhlopids, and the leptotyphlopids,
And I
or the
must say that those are
typhlopoids, leptotyphlopids.
the living examples of those early lineages. We have older lineages in
The
fossils.
extant, yes, exactly. I think that's kind of my point.
Really derived, right? They're
That
extremely.
is
They are.
exactly it, Ethan.
Yeah.
They are hyper derived. And many people until fairly recently as in within the last 5 years, many people have been arguing that they kind of represent the ancestral state for what snakes might have looked like, the state into which snakes might have gotten and then subsequently kind of diversified from there because of this silly question of, why did snakes evolve to look like snakes? Like, what was the thing that basically the question of why did snakes lose their legs? And I think there's more or less no ambiguity anymore. Snakes lost their legs because they were becoming more sinusoidal in their movement and were becoming kind of leaf litter dwelling probably organisms, because that's how skinks lose their legs. They do that, and then, you know, and skinks have done it dozens and dozens of times. So, but the problem is, if you start to look at the surviving lineages of the blind snakes, you start to have this, you can fall into the trap of thinking that that's what the ancestral snake looked like. But what I showed, and we've talked about this previously on the podcast, I had this paper together with Katie Strong and Michael Caldwell, where we talked about what did the ancestral snake look like, and how do we relate the blind snakes, these three different lineages of blind snakes, to that ancestral state. And the point was that they are hyper-derived, right? They do not represent that state at all. So if you look at the figures within this paper, you can see that those snakes actually don't match the rest of the snakes in terms of, you know, those early branches, they don't do the thing that all of, especially the colubriforms, are doing, because they haven't. They've gone a different evolutionary way, and it's probable that the early innovation that those three lineages underwent that protected them from mass extinctions, which is a very burrowing lifestyle, living on an infinite resource, which is, you know, the eusocial insects, that they don't really represent anything else that was around, but they're kind of the last living leaves of a very bushy part of the tree. And I think that kind of allows me to bring that quote that I wanted to bring up in here, which is, I think, this really fantastic point is so eloquently written. So if I can quote very briefly from the paper: "Perhaps most importantly, the pruning effect of extinction through the selective elimination of ecologically and phenotypically intermediate clades may exacerbate the distinctiveness of clades that comprise the survivors that we observe in the present day." I think that's such an important point. That's why I wanted to quote it, because we get this false impression of how distinctive the lineages are because we lose those intermediates. We lose the transitional forms. We are only informed based on our survivors, except for the fossil record.
Isn't that just generally good advice for understanding evolution
It
anyway?
is, but I think it's something that people forget all the time.
And they make a point here in the paper to point out that that is probably most likely the case in the evolution of many other clades. Like, for example, I was bringing earlier in our discussion that the evolution of flight in birds is something that happened many times within dinosaurs, at least 3 or 4 times, and it only took off with birds. Why? Probably the same reason. There was a perfect storm of circumstances, which in this paper they talk about, you know, it can be one or more unknowable events that could even, you know, it could even have to deal with something like geography, for example, or
environmental
climatic
factors of
events.
some kind. Yeah.
Yeah.
That's a really fascinating idea, you know.
And I think the other thing that they say here is that a lot of this is potentially unknowable, right? It might be that there are factors that really do make this perfect storm that creates a singularity. I think there's no question to my mind that birds are a singularity here. Snakes appear to be behaving as though they're a singularity. I'm actually surprised to see the statistics support that, because my impression has always been, well, snakes, okay, they're very diverse, but then taxonomically they're maybe a little bit given more attention than they deserve. And so, you know, and people forget how hyper diverse geckos are. And some of the other lineages that are kind of underappreciated. And when you've got a background of such heterogeneity and so much rate variation in terms of speciation, it is very hard on first principles at first glance to say, okay, snakes really are an exception. There's a bias, a preconception that would be the case, but I don't think if you're really informed about how these things work, it's not necessarily a given. So it's interesting to see the statistics really support that snakes behave in this singular way, as do, of course, the birds. And in fact, some groups of mammals probably also are behaving in this, like,
bats.
yeah, bats.
Bats are huge,
Yes.
hugely. I mean, people forget how diverse.
Half of all mammals are
I was
bats.
gonna say, isn't it like a
Exactly.
ridiculous number out of the total mammals? Yeah.
Yeah,
Yes.
like half of the mammals
Half
are
of all
mammals.
mammals are bats, which is
And
really
the other
funny
half is
because the other
rodents,
half
and
is
the other,
rodents. That's
And
it.
statistically, we're all beetles. So,
Yeah,
you know.
statistically we are all
Yeah.
beetles. But that again, statistically, not just are we all beetles, but there are specific groups of beetles that we all are because of the hyperdiversity of those groups. And
Yeah, right.
and parasitoid wasps is another one. Like something
Uh,
happened at the origin of parasitoidism that led to this hyperdiversification of this group. Orchids as well, like
it's
vast
like
majority,
it's like
most
this
plants are
is a
orchids if you
pattern that works, and
look.
that's that, I don't know, like you can hit on parts of the pattern and it sort of works, but when it's all there,
yeah.
I
And
like the fact
and
that the
the
papers
paper
kind
does make a point saying that it's non-predictable either. Like you cannot. I mean, how can you predict this? All these circumstances coming together to favor a
right.
certain clade, even though certain aspects of that is making that clade successful have been present in many other clades.
I love that this paper's basic thesis is it's shit's complex, man. It's just it's
I also
complicated.
like the fact that they combine. It's not just like genetics, or it's not just morphology. They've combined the two to be able to show that there's a lot of complex things happening, but support the fact that that's happening.
Right.
And honestly, like the way I like to think about it is you know in these different taxonomic groups, it's having the genes for plasticity, like
Mhm.
which I don't think gets discussed enough, because it's exactly the same thing. Like with the chameleons, it's okay. They've got all these different traits that are displayed in other lizards and things as well, but they have something special about them. Um, I mean, I don't know if there's anybody doing specific work on that,
Is that
getting those
is that a
genes.
thing? Like the studying the evolution of evolvability? Is that
Yes, that is a thing. Yeah. Yeah.
like why lungfish are kind of like I'm just a lungfish for several hundred million years?
Yeah, to some extent. I think, there are lots of different angles that you can access that from. So there is, for instance, plasticity specifically has receives quite a lot of attention because having phenotypic plasticity, which has a very strict definition that I don't want to botch by trying to give it, but it's essentially the ability to over multiple over I think at the generational level to be able to adapt to a different environment or develop in a
I
specific
can see the
way
YouTube
specific to a given
comments now
environment.
where they're like, Mark said the wrong
Yeah,
thing.
yeah, yeah. Yes, yeah. Get in the comments. We'll happily throw you under the bus in the next episode. No, no, no. But essentially there is the difference between flexibility and adaptability, and then phenotypic plasticity.
That's a really interesting idea. Yeah.
Yeah, and so ultimately there are cases where plants are the best example of this, where you have phenotypic plasticity in the sense that you can raise, especially tomato plants get this a lot, so you can raise a tomato under specific conditions that makes it very small, kind of dry adapted thing, but then you can take the same, the sibling plant from the same tomato and you can raise it in a lush environment and it will grow in a very different growth habit. So they're like plants in general have a level of phenotypic plasticity that animals can
And
only
that's
dream of.
all right. Not to go down a rabbit hole, but that's different. That almost sounds Lamarckian.
It is
Uh,
not Lamarckian. It is not
at all.
Lamarckian.
okay,
No,
Yes.
no.
yeah.
This is ingrained in the code. So the in the
So okay,
um
so conditions are this way. code
Yes.
goes.
Exactly.
Okay,
And we
I got
see
it.
this, there's fantastic examples from frogs. So there are specific frogs where when the tadpoles occur at sufficient density, a given percentage will
Cannibal.
become
Yeah,
cannibals. And this is
yeah,
Yep.
also phenotypic plasticity, the ability to deal with an ongoing environmental condition that triggers a developmental response that
got it.
that
Got it.
It
changes
happens in
slightly the
salamanders.
developmental program.
Yeah, I was going
There's
to say that
salamanders that have
you
this as
see
well.
salamanders, you see, I think also probably with neotenic
Yeah, you could
like
consider to some extent. I don't know if you can really consider the facultative metamorphosis to be a kind of phenotypic plasticity. I suppose to some extent you could. But yeah, I mean that is the gist of that
got it,
whole,
yeah.
and yeah, that's a huge field of study. Because you
And
know,
in a way, this paper is that field of study because they're using a lot of different, they're using, like Hiral said, they're using a wide variety of data to answer or try to answer or look into this question. They even have, like Mark said, an outstanding number of ecological data, which I think sometimes is overlooked when you're looking at this kind of answers. Like when you're looking at the kind of question, like why is this clade so diverse? And sometimes the ecological data is sidetracked a little bit. And I think this paper does a great job at not doing that.
Mhm.
and it's exceptionally difficult to gather. I mean, getting a large number of records together that are well collated, that are taxonomically correct, is surprisingly difficult to do. Not that we are vouching for the taxonomic correctness of the data that are associated with the paper. We haven't looked at it, but in general, if we have,
But judging by the authors that are in the
giving
paper,
faith in the authors and the
It's
article.
it
Um,
seems to me like it's a really objective, data driven answer to what is really kind of a wishy-washy, like very amorphous question. Like
mhm.
basically, why are snakes? And
Why
then
are snakes? Yeah, and well, I think that as I said, like at the first question here is, are snakes really so special? And I think that the data shows pretty indicatively that they are. And then the question of why they are seems to be a combination of right stuff at the right time, that happens to have gone well for this, for yeah, in the right place. Um,
yeah,
for the right for this particular period of geological time, you know.
so in a
So
way, basically like everything that happens in life, because when everything happens to you, you also have to be at the right place at the right time. So
yes.
it's, in a way, it's the same thing for everything.
Hmm.
it's yeah. I got this. This is another one of those things where it's like if you were to play the tape back,
Right.
you wouldn't necessarily get snakes again.
That is exactly it. Yeah.
Yeah,
And there's a great book by Jonathan Lossos called Improbable Destinies, that talks about this for the lay audience, and it's really interesting to think about it in those terms. And I think that that's the crux of any paper that's like this, is
yeah.
how much of this is predictable, because some of evolution is absolutely predictable given these conditions and this combination of traits, this will happen.
You're
This
almost
will
Knowledge
happen,
definitely
are a
or
perfect
or
you're
example.
almost definitely
extinction.
gonna get legless something. Like
Well, and
legless
knowledge are
snakes.
a perfect example of this. It's improving with how fast they evolve, like shorter legs because of
Exactly.
storms.
Which, to some extent is the reason that Jonathan Lossos wrote the book, right? Because anoles
Yeah.
really are a wonderful system in which to study
Yeah.
precisely these questions. Why that is the crux of why every one of the islands in the Caribbean has the same set of ecomorphs, is that when you play the tape again, yeah, they do the same thing because that happens to be a good way to divide the space that exists. And that has been basically proven in those organisms. So the question of how did snakes happen and why are they so much more special than all of the other? Because, as Gabriel has highlighted, um, there are a lot of different groups that have lost their legs. Basically, almost every group. Yeah. But even that figure, Gabriel, that you're showing
No,
from the
even that,
paper, figure 2, does not have all of them. And not even every family that has lost their limbs, because almost every family of lizards has lost their limbs. Obviously, chameleons are.
Notably, the exception is iguanians.
Yes.
It is like an outlier.
Yes. Yes. Which is quite unusual.
That's
So
another thing that would be worth studying, like why this group of lizards
I'd
haven't
like
done.
to see some legless anoles.
yeah, I mean they are the leggiest of the lizards, to be fair. So
Maybe
like,
it's well, with them it's like we evolved these fancy toe pads, but then I was like, no, you got Lealis
no,
on the
no. I
gecko
mean
side. That
geckos
yeah.
definitely lost legs. You have the yeah. I mean it really basically
There's got to be
everything.
some pressure to
I
keep
mean,
those toe pads.
Yeah.
Oh
amphisbaenians,
yeah,
how
yeah.
much legless you want to go?
Yeah, yeah. But I think, the iguanians really are an interesting case. So we have the true iguanids, we have the agamids, we have the chameleons, all of these groups who have not
I
lost their limbs. But then I think one of the things that really is remarkable in those two, like the agamids and the iguanids especially, is how much their locomotion is powered by the hind limbs.
yeah.
And then chameleons are special because their limbs are so hyper specialized. So
I think
like chameleons do the least sinusoidal of all of the movement of all
yeah.
lizards. And then I think a lot of it could be a hind limb motivation, hind limb locomotion thing that really sets apart the agamids and the
I
iguanids.
My money would be on marine iguanas. I know, we're not into the evolving leglessness through aquatic, but
Yeah, that doesn't happen. So don't even
Doesn't
bring it up
happen.
because
No,
it doesn't.
I'm
You're
gonna
opening a
stir some
can of
sh-
worms
mosasaurs.
I'm gonna stir some shit, and that's my money's on that. We'll come back in a few million years.
Yeah, we need some fossorial agamids to evolve. That would do the
Oh,
trick.
maybe yeah, maybe like uh Chuckwalla or something.
Right. So I think we've done this paper some justice. It's a very interesting insight, of course. Um, Important to see the snakes in the context of the rest of the lizards, and I think that's a refreshing thing. And again, the fact that what the lizards have or what the snakes have done is kind of a perfect storm is nice because it is a pointer toward like perfect storms are a thing in biology. They do somehow opportunity arises and the right thing is there at the right time in the right place to do the thing. And so, yeah, what I wonder is if you'd been there at the time, in the early days, imagine to put yourself in that situation where you're kind of walking around and your day-to-day fauna is this group of early derived snakes. Would you have been able to tell? I don't think so. You'd be surrounded by these
Oh,
big fuck off monsters that are also walking the earth with you. And you'd be thinking to yourself, oh, those little piddly things, there's no way they're going to survive for the next 100 million years. And then lo and behold, here you are.
I mean, but you
So,
can tell that to the gecko more like the geckos in the Jurassic, saying
absolutely.
these little lizards are, will this clade for some reason stay all the way until the
Yeah.
modern times? Yeah.
Yeah. And I think that's really important. That perspective is quite important because thinking about what is going to happen next from here, we don't know which of the lineage that's around today is going to survive for the next 100 million years and what the potential geological record might look like if you were to reflect back on it from that time point. So,
And the
uh,
circumstances, the perfect storm of circumstances that will determine one way or the other are non-predictable
yeah,
and are basically we're not.
luck. It's luck.
Yeah.
it is kind of chance.
Yeah.
Yes. I think, as I said, like there is contingency here, that's luck, and there is determinism. So there are certain things that under the right circumstances will always go the way that they go because of the way that they go. Limb loss is a really, really good example. So
there are certain things that we can predict, but I don't think that we can predict at the level of the, the big picture. And on that note, I think we should probably move on to talk about two other papers, snake papers, that we just wanted to mention because they are interesting perspectives. Let's perhaps talk about the Madsoids first, because that does seem to be kind of in keeping with the theme, what might have been. So, Gabriel, can you give us a
Also,
brief
where they are
rundown?
located.
Yeah, so can you give the listener, we've just been talking about modern snakes, modern snakes, modern snakes. Can you give the listener a brief introduction to Madsoids? Because if they're not paleo people, they may never have heard of them.
You want me to do it?
Sure.
Oh, so Madsoids are a group of an extinct group of snakes that lived mostly in Gondwana, but there are some examples outside of it, and they are known for having achieved very large sizes. If you saw Prehistoric Planet and you saw the episode of Madagascar where there's this snake preying on a small mammal, that snake is actually Matsoia itself. That's the genus of the one of the many species of Matsoia. I think it's Matsoia madagascarensis. And I did the concept art for that species, for that one. And Matsoids were able to achieve very large sizes. And one of the things with this paper and this new species that has been described based on some very, very large vertebra is that this Matsoid could have been one of the largest snakes. They say that it could have rivaled in size even Titanoboa, which is the largest snake known so far, which is from is it the Eocene of South America, northern South America? I think it's, or Paleocene. It's very early. It's right after the extinction of the Cretaceous extinction at the end of the Cretaceous.
It's always portrayed as just a giant anaconda.
It's a boa. It's a boa. Yeah. It's a boa. It's a boid. It's the largest snake so far. I think it's measured to be like 12 meters or so. But this new species, which is what's the name? I forgot the name.
Well, let's first talk about the paper. Introduce the paper. Just generally, it's by Datta and Bajpai, published in Scientific Reports, and the title is "Largest known Madsoid snake from warm Eocene period of India suggests intercontinental Gondwana dispersal," and the new species is called Vasuki.
Oh, yeah.
Vasuki indicus.
So for those that are interested, Vasuki actually means kind of king of snakes. So they're basically talking about it and as the king of snakes.
That's a very nice genus name.
Discovered. Nice. You had family near where it was discovered, you said?
Yeah. I mean, yeah, my family is from Gujarat, so it's quite interesting to think, if I'd stayed in herpetology and gone, stayed in India, what might have been.
Yeah. Yes.
It's one of those perfect storm situations again.
Yeah.
Yeah, so the new species is it shares with so many of its so many other fossil taxa the fact that it is comprised entirely of vertebrae.
Yeah.
Right? So loads and loads and loads of snakes are described exclusively from their vertebrae.
Well, let's just first say that snakes don't fossilize well at all.
Mm-hmm.
The fossil record of snakes is very limited.
They tend to fall apart.
They tend to fall apart, and like especially the heads, we have so few snake heads in the fossil records. We mostly know them from cervical or dorsal vertebrae and stuff.
That's the most substantial bone in them, right? Because the skulls are just super delicate things, even on a large snake, right?
Yeah.
Yeah, I think that's exactly it. I mean, the snake skull is anyway, it has that high degree of modularity and mobility, and that leads to a relatively easy process of yeah, falling apart. And which is quite interesting because I don't know how people then do reconstructions of these silly animals, because the head is so much of what people want to visualize, and it's very hard to reconstruct an animal just from a vertebra.
You have to like basically see where they where they fall phylogenetically and try to make the best out of it. That's basically what you've
Which is
done.
particularly different in a family that doesn't have any modern descendants, where we don't have, I don't know, do we have a single skull from any Matsoid?
I think we do. We
Okay.
do. We have. Thankfully, we have like I think there's one or two taxa that have skulls somewhat well preserved, because the good thing with Matsoids is that they almost reached us. They were extant until the Pleistocene. So right a few thousands of years.
By that much.
Yeah, a few thousand years ago.
Uh,
So
is that why? Is that why like Titanoboa is always reconstructed to look like it's like an anaconda? Because basically, that's the closest large-bodied relative
well,
But
that
it is a
exists.
true boid, so
it
that
is a
is,
true boy. Yeah.
yeah.
With boas, you cannot really go that. I mean, there's an anaconda is not that different from a boa. I mean,
Right.
you have definitely also
That's what I mean. Is
inferring by
they're
the
just
fact
they're
that
ballparking it
Titanoboa
based
must
on
have been semi-aquatic. Chances are that it kind of looked like the semi-aquatic components of boas, like boa itself or eunectis, which is anaconda.
so
So
sticking close to Unix is the probably the
well,
right answer.
I would say sticking close to boa and eunectis.
Yeah.
I would, when I reconstruct Titanoboa, I try to, because I think it's been reconstructed too much to look like an anaconda, and I
That's
don't
what I
have
was
to pay
getting
for
at.
that.
Yeah, yeah.
Yeah, but I mean, in reality, you also have to take into consideration the lifestyles of these animals. You also have to keep that in mind when you reconstruct them, you know. And in the case of Matsoids, it's the same. These are animals that were huge, and they, because of their place in the phylogenetic tree, which is another thing that this paper shows, is where Matsoids are located at. They're actually quite basal snakes in a way. I mean, they're closer to stuff like uh, uh, anilids, which are today only represented by a single species, the coral pipe snake.
Which is a weirdo, also in terms of its skull anatomy.
And it's also very derived, because
Mhm.
it's an animal that is highly fossorial, and
Yes.
So again, a bad example of what
so it's a bad example.
integrates.
But you can make certain extrapolations of what those early snakes would have looked like.
Definitely.
Yeah. I find that really interesting with what you do with the paleo art stuff, because it's basically that's the art part is making this like educated guess
Yeah.
as to what it might have looked like.
Yeah, that's important. And one cool thing about this new species about Batsuki is that Basuki, sorry, is that it would have reached such a large size that I don't know if they're thinking that it could have been semi-aquatic as well. I don't see an animal that size not being semi-aquatic. A snake that size kind of had to be semi-aquatic.
Well, I mean, they used to say that about sauropods, so you know, yeah.
Oh, but it's different. It's
It
very
is
different.
different. Yeah. I know
There's no air sacs in snakes.
Yeah.
Well, there might have been in the soft tissue, but yeah, I think that it's I think there's a decent chance that it would have been an aquatic
Well, they actually
giant, like,
said that I'm actually reading that they actually said it's probably not an aquatic animal. Its vertebral morphology, this is from the Wikipedia page.
mhm.
Its vertebral morphology instead suggests a terrestrial, possibly semi-aquatic lifestyle when compared to the related matsoids and modern pythonids. Batsuki fossils were deposited in a back swamp marsh. Well, I mean, that
Now,
is,
yeah.
they are saying that it's some, well, they're probably considered that anacondas are super aquatic, and they are, but in reality, they are semi-aquatic.
Semi-aquatic, yeah. I mean,
Yeah.
yeah.
We know it wasn't a
Yeah.
gliding animal, is what.
Definitely not a glider. Yeah. Or if so, then only very briefly.
Yeah, but maybe that was its hunting method, was it fell out of the tree on you know.
When you're
This
considering a huge snake that was between 10 and 15 meters long, I don't think that would have would have moved too fast on land at all.
It
yeah.
would have just crushed its prey to death immediately. You know.
But of course, Matsoes are kind of a mystery in many, many, many respects. So
Yeah.
But yeah, an interesting, it's so cool to have another giant on the field. I mean, it must be cool to work on a vertebrate that's not like teeny, teeny, tiny when it comes to working with fossil snakes. So that's probably quite pleasant. And if people are looking in the right area with the right techniques, they might find, more substantial pieces of the body. Although I must say that the body of snakes tends to be fairly unrewarding in terms of like, unless you find the head, it's uninteresting. That's
I didn't know where you were going with that for a second, and I thought you were gonna say it was like could be out there. It's a
oh
cryptozoological
no. Definitely not. Definitely not.
you know,
Yeah,
start
so
a megalodon conspiracy type thing.
Because we forgot to say that Vasuki is from the Middle Eocene.
yes,
Okay.
Quite a long time ago, from the Middle
quite
Eocene,
quite
India.
dead. Quite dead. Although I don't know how old megalodon, the oldest megalodon
I think
fossils are.
closer. I think it's
Yeah.
closer in time. Yeah.
No, Megalodon is like the oldest, the youngest one is like, I think, like 2 million years ago or something like that.
Uh-huh.
Yeah.
Well, could still be out there. No.
No, no, please don't say that. It is not
The
out
Some,
there.
Meg.
somewhere
For,
David Shipman is like,
for
oh,
all of us that have to deal with people in the
Ah,
paleontology,
no.
that have to deal with
Okay.
those annoying questions.
All right. I'll cut it. I'll cut it. Uh, God. Yeah, no, it's really unforgivable. Don't do that. Don't be that guy. Um,
No.
yeah, shall we talk about the last paper?
Yeah.
Yes,
Yeah.
Tropidophytes.
So let's talk about some very poorly known boa-like things. Specifically, these are the tropidophids, which are not boas,
Dwarf boa. Yeah. But
but
are not
are called dwarf boas for whatever reason. The paper that we're talking about now is called Molecular Phylogeny and Biogeography of the Dwarf Boas of the Family Tropidophidae, Serpentes alaphidophidia, published by Hussam Zaher and colleagues in Systematics and Biodiversity. And this paper is really just a treatise on the tropidophids and kind of an update on the overall overview of them. So,
whole family. And let's say that they have a weird biogeographic, you know, distribution. Like they have a weird distribution because they're distributed from Central America and the Andes, west of the Andes in South America, and then in the Atlantic forest of Brazil, and then in the Caribbean, in the Antilles, where they are very diverse there.
right,
That is
yeah.
weird.
They are certainly weirdos. Yeah, if I'd thought about it earlier, I could have gotten a specimen. I could have shown them off. But alas, yeah. So really, this is just kind of an update on the things. It's not like, I guess it's not going to be super world shattering for most people or world shaking for most people, but there are some relatively important takeaways, not least of which is the loss of a genus, because Trachyboa is reconstructed as folding within Tropidophis, and so to reconcile that situation, the simplest process is to unify Trachyboa with Tropidophis. So the two species of Trachyboa are synonymized or are moved into Tropidophis in order to.
You want to talk about why that approach and the difference between splitting
Yeah.
and
So
lumping?
yeah. So basically at this point you could divide you could cut off in order to, if you wanted to protect Trachyboa, because it's relatively deep within the tree, you have two options. Your option one is to cut off everything that's above Trachyboa in the tree and move it into its own genus or its own genera, their own genera, depending on how many branches there are. In this case, it's really just one. So there are two species that form a single clade, which is Tropidophis Kakwangoe and Takjanovskii. So those two species form a clade that is sister to all of the rest of the group. The next branch down the tree, if you make the tree into a ladder, the next branch down would formerly have been Trachyboa, and then you could keep going, and then Trachyboa is sister to everything else within Tropidophis. And so you have two basic options here. Option one, you would split off that first deepest clade, keep Trachyboa, so you'd have one new genus, keep Trachyboa, and then all of the rest of things would become Tropidophis. Depending on actually which lineage holds the name-bearing taxon for Tropidophis, which I don't know which taxon that is. So it is in theory possible that it would be the outermost thing, and then everything would have to change to a new genus except for the outermost clade, which would be very inconvenient indeed, because there is something, there's a paper published by Miguel Vences and colleagues in, I think it was published in Zootaxa back in the day, and it's called Criteria to Promote an Economy of Change for higher taxonomic groups. It has a longer title than that, but that's the gist. And in that paper, they advance what they consider to be primary criteria and secondary criteria for what action to take when you're faced with a dilemma like this. And basically, they said there should be an economy of change. There should be a consideration of how much is this going to upset the existing taxonomy? How much is it actually useful? And under what circumstances is it kind of justifiable to give something its own distinct name? And what it boils down to is, okay, there's no hard, fast, clear rule. Often these things really are going to be a matter of taste. But in many cases, if you look at how many changes would be necessary, you can take what I have published as in a separate paper as being called a parsimony of changes. The minimum possible number of changes would be the most parsimonious solution. And so what you ultimately want to do is take the change that changes the name of the fewest number of taxa. So if you're faced with where everything else is equivalent, sometimes it makes sense to do a bigger change if it's really justifiable based on, morphologically distinctive features. So in this case, you're faced with either moving two species to a new genus and creating a new genus, or moving two species from an existing genus into another existing genus. Both of those are kind of one step. Well, actually kind of two steps because you're moving two species. But in reality, the merging thing is less of a upset to the taxonomy than making a new genus, because making the new genus creates an additional name. It creates more, more chaos. And so in this particular instance, the most parsimonious thing was to move Trachyboa into Tropidophis. Under other circumstances, you might have weighted it a different way. Dividing further is typically not advisable under a circumstance in a tree like this, because it can create relatively arbitrary units, unless there's very clear distinction between those units, which there often isn't. So yeah, I think that's the gist of it. Is that what you're going for, Gabriel?
Yep. That's a good way of explaining to people how do you make the decisions.
So it is, and I think what's important to know is that this is kind of an arbitrary thing. Like you do have to take those decisions, and some people have a philosophy of more splitting is better. It's better to have more units. Other people are more conservative, and you can solve this with an intermediate solution. But sometimes that intermediate solution is also not, parsimonious or is not economical. So, for instance, there would be the solution of creating 3 subgenera, one of which is Trachyboa, the other one is Tropidophis, and the third one is whichever the new lineage is, which actually creates more structure, which can be a better reflection of the actual diversity and can help with the management of the units without oversplitting the genera. But then, in reality, is maybe not better than just throwing Trachyboa in there, because I don't know what the traits were that distinguished Trachyboa so strongly from Tropidophis. Because to my mind, they look pretty similar.
I always thought the philosophy here was, how many nerds can I upset?
No, you try not to upset the nerds. I think that's part of it's actually part of the process.
But in some cases, you will, and there will be bitter fights about it.
I'm still mad about
when they try to divide anomalous. That's what happened.
Oh,
it's
yeah.
Famous.
So there's a whole debate about Bufo and some other cases that I probably should never talk about on the podcast.
Rana.
But so Bufo and Rana, where
you can take Rana from
I can do anoles because actually anoles are one of the best examples of where the lumpers have won. And it's really kind of frustrating for those of us who want to actually have usable units. Because the problem is that the former family Dactyloidae, which is now Anolidae because someone found out that the name Anolidae was available and was an earlier name. So the Anolidae, the family Anolidae, is equal to the genus Anomalous. And all anoles, chameleon anoles, twig anoles, every island anole, every continent anole, all of the anoles are today considered one genus Anomalous, which is to say one family Anolidae, which is to say there is absolutely no structure or utility whatsoever in higher classification within anoles, which is ridiculous because there have been multiple, I think, well-reasoned and well-justified proposals for divisions into Norops and other units, which
and those
are
groups
super helpful.
And they've been separated for
100 million years.
Yeah, not so much, but several million years.
Millions and millions of years. Yes, many millions of years. Okay, maybe not 100 million, but it's a lot of years.
like I think it's something like 35 or something, 35 or 40 million.
Fine, I was off by half, but still, it's a very long time, and there's no reason to lump everything together, except if you're too lazy to learn another. Sorry to be that blunt, but except if you're too lazy to learn another, because it does divide things up. And if you say, okay, well, there's nothing that's like super distinctive between these different things, that's not entirely true. As far as I understand, all of the all the mainland anoles stay in Norops, and everything on the it's the other way around, right? All the island anoles are.
There's no geographic definition. There is Norops and also what we call alpha anoles, which are the other group. But Norops, it's very different because they do have there's some structure in the tail in the caudal vertebrae, which is completely different in Norops and the rest of the anoles. So even that distinction you have
easy peasy. Yeah. So, it's bizarre to me that the anoles are lumped the way that they are.
I'll go with you on that one, because I've always thought anomalous was
it's a waste bin. It's an absolute waste bin. We've got everything. Okay, yes, there is one large monophyletic group, but it's
literally for that.
But it is a large, totally useless monophyletic group. The whole point of higher taxonomy is utility. The only purpose that it can possibly serve is to help us manage the units in our heads, right? That's all it does, because biologically it has no meaning. Biology cares about species.
The anoles don't care.
The anoles don't care, right? It cares about species. But for us, it helps us to manage the units when we know what genus we're talking about. And we use terminology, a part of the taxonomy, in order to manage that. And that, by the way, is the same, that same thing applies to the rana, it applies to bufo, it applies to various different,
I'm
because
just being a jerk.
I know that it's inconvenient, and I think a lot of people find that it's inconvenient to have to relearn names because it's easier to talk about Rana Catasbiana and Rana Woodhousey and Rana blah blah blah.
Well, there's a sort of nostalgia for it, I guess. It's it's like that's
Yes,
what
and people are stuck in the mud and unable to update their definitions, or it's inconvenient to them, or it means that they have to relabel all their files or whatever. But it when it's at that scale, as it was with Rana and it was with Bufo and it still is with Anoles, it's at the scale of being vastly less convenient when it's lumped than it is when it's
split. Really
split.
need to use it.
A good
For the
case
people
of
who
that
use it.
was a good example of something that was very informative, to the point that some of those things ended in completely different families, was Eleutherodactylus.
Yes,
which
I thought you were going to say Hylidae, because it's also the same, right?
Hylidae was also the same, but Eleutherodactylus was a genus that was hugely diverse.
Yes.
Like 800 species, and some of those species ended up being in different branches of
now
different
it's now it's Pristimantis that all used to be Eleutherodactylus, and now they're the waste, like the giant hyperdiverse thing.
It is the advent of genetic understanding genetics better, right? I mean, that's
A lot of it had to do with getting an absolute answer to the question that we didn't have before, because before there was just morphology, or morphology plus bioacoustic ecology, etc. But there was
no way
all
to
this
tell
data, but
convergence
no strong indication of what actually were the grouping factors. And now it's easy to go along the tree and start to prune at the places where it's both convenient and obvious to prune, to create manageable units. But it also, and I think this is the crux, by thinking the other way around, by looking at the genetics and saying, okay, these are lumped together, what are the features that they share? Thinking that way around rather than, oh, these are the ones that seem to share this feature, which is very easily misled by, this is what happened in the SquaMate phylogeny, where the Dibamids were in completely the wrong place for the longest period of time because they're legless, and so they're being lumped with other legless things. The genetics came along and was like, no, Dibamids are sister to everything else, or sister to geckos. That was a huge revelation, and that was because everything had been misled by the morphology. And I think that's the same thing that happens in a lot of these cases. Like, we use non-natural grouping features early on because that was all we had available to us. Now we know the natural groups, and we can use that in order to find out what would be the best way to split these and what features do they actually differ with.
As an artist, and Gabriel, maybe you can back me up on this, but it when you do art, when you draw, you always go from very general sketch to more specific down the road. And I feel like that's what we were seeing here. Like, the early phylogenies are sketches, and as we know more and we observe more, we get more refined, more specific.
Yeah, and that is science in general. That's how science works. You start understanding something and you refine it, and build on your previous research. Build on previous research, build on previous research until you try to, get a clearer and clearer picture. That's hopefully what we're doing.
And I think to maybe stretch that analogy further than is perhaps natural, if you're resistant to changing some of the lines in your drawing, it actually makes the drawing worse for a long time.
Or not as accurate, at least.
Or not as accurate. And if you finally go in and fix the bloody lines so that it is the way that it should be, then you get a better picture, right? And I think that
you are correct. You are objectively right. I just Anaxarus doesn't have the same charm.
It doesn't, but it is a little bit more practical. And I think, one of the unfortunate things that has happened along the way, so a lot of the higher taxonomic changes within the frogs especially were enacted in a 2006 paper by Daryl Frost and colleagues. It had enormous consequences for the higher taxonomy of frogs. Subsequent papers have continued along those lines. This, by the way, the same thing happened with Colubridae when Colubridae was split up. But to talk
They're still
specifically
resistant
about
to that too.
Sorry?
They're still resistant to that too.
Of course they are. Of course they are, because it's more convenient to have a leaf be everything rather
Yeah, I
than
was gonna
these
say
individual
it's a
units.
leaf,
Yeah.
isn't it? Yeah.
Yeah. But when you have with the frog, transition that came along with that, that was a phylogenetically informed paper that started to make all these changes. Unfortunately, the way that the changes were made in that paper left many of the new genera without morphological diagnoses. Now, it is possible to diagnose them morphologically if you go and do the work and you find out what are the traits that actually differ between these different things. But in fact, in the subsequent 18 years since that paper was published, many of the groups that were established still haven't been revisited from a morphological perspective to actually figure out what are the traits that differentiate them. Which leaves
That's
people
why,
like,
because morphology work is hard and
it's
nobody
hard
wants to do
and
it.
time consuming and complicated and requires, especially at that level, requires an availability of specimens and a knowledge of anatomy that maybe a handful of people in the world possess.
Well,
And
it's
so,
also maybe some of the sour grapes over just changing the names, what feels like willy-nilly because you haven't backed it up with the morphology.
But
sure.
can I say something really quick? And this is something that it struck me when I was doing, taxonomic work based on morphology. It really helps to be an artist because
Mm.
we have a detail-oriented eye that other people might not have, and we get to catch morphological differences that other people overlook
Yeah.
sometimes.
that.
So I've found myself finding out, well, don't you see this difference? Like, don't you see? It's like, oh yeah, it's like, yeah, it's very clear, but it's overlooked by many people. So it does help. So many artists should go to work into taxonomy because it's really helpful.
But so one of the things that, I think that we need, or, maybe let me phrase it a different way. One of the problems that I'm faced as a curator here at the Natural History Museum is that there is a lot of uninventoried material. And getting identifications on that material is the barrier between getting it before getting it into the collection. Because until now, there was a stupid system where you had to know the family something was in order to give it a number. I solved that problem. But in general, it's not very useful for me to inventory things as frog SP, right? So what I do want to do is get it down to a genus, if at all possible. But the absence of a generic key to Anura does not exist. There isn't one. Makes life so unbelievably hard. Because many of the groups, like, honestly, if I had an unidentified frog without a locality in my hand, I can't tell you if this is a mantellid frog from Madagascar or a strabomantid frog from Central America. Wouldn't know what the traits are that differentiate those two groups. They are super deeply divergent.
How we didn't know it was that bad?
It's that bad. It's that bad. We don't have characters for Ranoidea, which is most of let's say, the old world frogs, and a lot of the Hyloidea, which includes the hylids, the bufonids, etc. There are some groups that have unique features that really set them apart. If it's got very prominent parotid glands, it's a toad. Cool. Bufonidae. Nice. Easy. But if it's a little pristamantid rain frog, the Malagasy frogs have converged on that really strongly. And I don't honestly know that I would be able to tell the difference because
is it something that you could, I mean,
there are traits,
yeah.
But often they're osteological traits. So
you can't take a genetic sample of that.
Not if you're willing to spend $200 per specimen. But I've got 6,000 specimens.
You have to be lucky. Huh?
Yeah, and you have to get lucky if they've been sitting, and most of these have been sitting on my shelves since 1914. So
So that's kind of a problem with the preservation method, right? Yeah.
yeah, well, it's a problem with the preservation method. We didn't anticipate that genetics would come along in the first place. But it's also just time. Time destroys DNA. Doesn't matter how you preserve them. Over time, even things stored in -80, not clear how long they will actually fare. Okay, they'll fare a very long time if they're kept at
And
-80.
you add to that it currently, morphological work is not being
It's not prioritized.
it's not being prioritized. You add to this, and then it starts creating this kind of problems. We have a huge problem with this.
Yes, especially like animals without locality. Snakes is the same. We have a whole bunch of snakes, zero locality data. And as a consequence, not only are they largely useless in the sense that, like, I don't know where they're from and therefore they're not really useful on a map or anything, but I can't identify them because I don't know that there is a key to the families of snakes or a key to the genera of snakes. Like,
you need
doesn't
like a
exist.
You need like a citizen science website, like they did for like the there was one years ago where you would identify possible planets because they would traverse the star, right?
Yeah, right.
You could go in and then it would you need to like put all these pictures up and have people take a stab at
Yeah.
identifying them.
And, if we have a location, often it's easy.
Yeah.
So for instance, today I looked at some frogs and I was like, it said that they were from close to Sydney in Australia. Well, it didn't say Australia, but it said near Sydney. And I looked at the frogs and I was like, that looks a little bit like a frog that I've seen before. And so I went into iNaturalist to the area around Sydney and looked up frog. And then actually I was a little bit more specific than that because I had the sense that they were a pseudophrine, but I didn't know what pseudophrine were called. I couldn't remember the genus name. So I was like, toadlet, Australia toadlet. And so then I found, okay, this is probably pseudophrine. So I went and narrowed it down to pseudophrine in the vicinity of Sydney, and then I came out with the identification. I was like, okay, cool, this is Pseudophrine australis.
I'm pretty terrible at identifying. I mean, I know the animals in my area, and then beyond that, like people will ask me, what's this salamander that I saw or something, and I'll be like, okay, where do you live? And then it's just, okay, salamanders that live in California, right, or Northern California.
Yeah. I confess that I'm particularly bad at that. So on Tumblr, I get a lot of people asking me to identify toads, especially.
Yeah.
And I'm like, okay, well, if I don't have locality data, I can't really help you. And then in many cases, especially for the two very common Anexurus species, I'm like, it's got to be one of them.
And,
so
let's say that, not everybody, you're going to become specialized and you tend to become specialized in certain groups. You cannot, it's impossible for one person to know every single thing. Right. But when you become specialized in one group, you're able to tell sometimes even what locality certain things are. Right. Like, if you ask me about Nemidophorus, I can tell you what locality certain populations are. And that happens to everybody. It happens to you. Breeders do that a lot with like.
Oh yeah, they get obsessed over it.
Yeah,
stuff like
that.
Yeah. I just want to go back. Like I really like your point about artists and in recognizing morphology. I think I'm always reminded, especially when I have talks with you, Gabriel, about how there is way more overlap between artists and scientists than I think a lot of people think. And and it's something that I think a point that should be stressed, because when I was growing up, that was not the perception that I had at all, and was kind of told, well, you're not smart enough or mathematically inclined enough to go into science, so draw your little drawings. And
no, it is crazy the amount of overlap. The amount of overlap is crazy. It's crazy. There is a lot of overlap, and I think that what you say is true. And I've heard this discussed in many other podcasts too, of many scientists that say like, yeah, it is. There is a lot of scientists who are also an artistic side, and they notice, the merge of both things can be, not only it happens, but it can be extremely helpful. Like I said,
Yeah.
if it can help you a lot in both sides, both in the artistic side and in the scientific side.
I think both professions are also extremely underpaid and undervalued too. So that's also
Unfortunately,
Certainly.
yes.
Okay.
So with the worst of both worlds, Ethan.
yeah, right. I have two careers that didn't pan out. Yeah.
Exactly.
Yeah. All right. Well, friends, I think we're going to leave it there unless any of you has anything last that they want to add.
That's fine.
No, I'm
Very good. All right. Well, thank you everyone for listening. We hope you've enjoyed this snake chat in its many different forms. Gabriel, where can one find you on the internet?
I'm at Serpent Illus on X, Twitter, whatever, Blue Sky, and I'm on Gabriel Ghetto Art on Facebook, and of course Serpent Illus on Instagram, which is kind of my main platform right now.
Hiral, how about you?
I am at Hiral's Hive on Instagram and at Hiral925 on the other platform.
Excellent. And Ethan?
I'm at Black Mud Puppy pretty much everywhere, and I'm on Blue Sky as well, probably more than anywhere else right now.
Wonderful. You can find me at Mark Scherz, MarkScherz, on most of the platforms. You can find the podcast at SquaMatesPod@gmail.com. You can send us your questions, thoughts, feelings, pretty lizards, all of the wonderful things that you are
fan art
enjoying. Fan art, always appreciated, definitely. You can follow the podcast all over the internet at SquaMatesPod. You can read the extensive show notes, including references that we've talked about, SquaMatesPod.com. And as we say on the show, Hakuna Squamata! Right, that was okay. I thought that was pretty good.